Hi, this is Miss Litton, and we are, say hi, you guys. Hi. Still period 1. And we are continuing our discussion about the cell cycle and cellular reproduction, and we are in section 9.2, the eukaryotic chromosome. And we're going to start off by talking about chromosome, and I'm going to blend this right into 9.3, chromosome structure. But I want you to see in this diagram, this biggest part here is the nucleus. What's this dark area right here? Nucleolus. Nice. Okay. And there's heterochromatin and euchromatin. Heterochromatin is the dark. Hetero means different. Dark staining. This is the non-active DNA, where this the patches where you see that are dark. Euchromatin is the active DNA. Active DNA. So both are a part inside that nucleus. Now, why would some DNA be active at one point, not during another point? It could be. Go ahead. What?
It's in the S thingy.
Right. Just a second. It's talking. Whoa! It's talking to me. Okay. Sorry. It could be depending on where it is in the cell cycle. Yes. Yes, absolutely. This could vary within your day. For instance, in the morning you have cortisol, which is your wake-up hormone. And light will help you start to secrete cortisol. So you're like, yay, I want to greet the day. I want to learn today. Let me start off by thinking of one thing I'm thankful for before I get out of bed. Changes your whole day. Okay, and then you get up and you start to proceed through your day. So you're not going to need that as much. At night, you're not going to want cortisol. You're going to want melatonin so you can go to what? You can go to sleep. So it can change throughout your day. It could change throughout the week. It could change throughout the month. It could change throughout your year, throughout your life. You'll have different DNA that's active at different times. When a cell is getting ready to divide, whether it's active or not active, it'll start to coil up, and it coils up into these rod-shaped chromosomes. Now, when we first see chromosomes, they appear doubled. And that's because when they first appear, you're getting ready to go into mitosis. So they have already gone through, what stage are they in right before mitosis? Right before mitosis. G2. Which means they've already gone through what? S. And S is when they doubled. So when the chromosomes appear, they appear as doubled chromosomes. And the part that holds the two copies together is called the centromere. We will talk about the process of DNA replication. That's going to come a little bit later. But right now, I want you to know that you have two copies of information, and the goal of nuclear division is to get one of those copies in one cell and one of those copies in the other cell. Okay? So how does it form this chromosome structure? Well, here I want you to see, you're really close up right here. You're seeing the double helix of the DNA. Yes? You got it? Okay, so there's your DNA strand. Now, if we backed away from that DNA strand, you wouldn't see both halves of the nucleic acid. It would just appear as a blue line. Do you agree with me on that? Okay, so now it's like I'm backing away, and that is the DNA double helix right there, the blue strip you're seeing in the middle. And the DNA wraps itself around what it's called histone proteins. And these histone proteins are all pretty much the same. There are both histone and non-histone. Okay? Histone is the standard, and the DNA wraps around, and then several of these histone proteins with the DNA, they wrap together and they form what's called a nucleosome. So DNA wrapping itself around the histone, then they start to wrap around themselves and form this blob right here that are called nucleosomes. Now let's back up a little bit more. The nucleosomes all start to kind of twist and turn and wrap on each other, and then wrapping even more on each other and wrapping even more until you're in this chromosome. It would be kind of like if I took somebody's hair and I just took 3 strands, literally 3 pieces of hair, and I braided those. And I went through and did all that. And then I took each of those and braided those together, and I took each of those and braided those together. I took each of those and braided those together until I got one big hunk of braid, right? It's braid on braid on braid. This is twist on twist on twist on twist until you get this rod-shaped chromosome. Now, I'm going to tell you, that's amazing to me. It's amazing to me. I have a necklace I haven't worn in 5 years because it's twisted, okay? And I cannot get it. And it's currently at the oh, I get to pick it up. It is currently at the jeweler's right now because I can't manage to untwist it. Okay? Your DNA, every time your cells replicate, it can manage to organize itself, and not only that, organize itself into sister chromatids that can be separated from each other in order to form more cells. That's pretty good, right? And you don't mess up that often, okay? And if you do, you'll get it fixed. So, without going to the jewelers. So on your 9.2 on the basics, chromosome structure, following S and G2 stages of interphase, cells prepare for mitosis. Chromatin, which is DNA and what? Proteins start to coil up into two rod shapes joined by a centromere. Okay, blue tells slate, why do you have to have two copies of the DNA? Okay, and the organization, DNA winds around, what do you call those protein spools? Histones, good. Histone protein spools that form a nucleosome. The nucleosomes then begin coiling until condensed into a chromosome. Slate, tell them the way the chromosome appears during interphase. And then blue, tell them the way it appears during mitosis. Go ahead. All right, this is going to come up in later chapters when we talk about control of gene expression, your DNA. Histone proteins are the standard. It's like, you know, it's the worker you just wind the DNA up on. Non-histone proteins, these are proteins that are regulatory. Remember how we saw there was that RAS, cell membrane protein, which could be stimulated by a growth hormone, which then phosphorylates this, which phosphorylates this, and eventually the cyclin levels rise, stimulating the kinases, and you do cell division, right? So when you have something that's controlling the cell, it's regulatory in some fashion. When it's allowing a DNA to be transcribed and translated into a protein or not, those are non-histone proteins. They are regulatory. Histone proteins are your standard proteins that are about the structure of the chromosome.
That was me.
It's up on the board. Okay, go, go, go. All right. So when you have the two copies together, they're called sister chromatids. But when you separate the sisters, they become their own chromosome. Okay, they like graduate to chromosome status. Kind of like your sisters when you're living in the same home, but when you each have your own home, you're called your own families, right? Think of it like that, okay? So that's what goes in B. Mitotic stage, chromosomes form and appear as doubled sister chromatids, held together by a centromere. All right, now let's talk numbers. This is transitioning into 9.3, mitosis and cytokinesis, the numbers. Okay, yes. You want me to stop it? Okay. We're stopping right now, so sorry.